Mindsetters, welcome to today's Grade 12 Life Sciences show. Today we have a special revision lesson in which we'll be doing exam questions for paper 2. We have selected highlights from lessons shown earlier in the term to help you revise this term's work. So please post your comments and your questions on our Facebook page or on Twitter @LearnExtra so we can help you with your revision. You can also download the show notes for today's lesson from learn.mindset.co.za or click on the link on our Facebook page. Now it's time to get on with today's lesson.
Now we start with the actual questions from the paper. This one here comes from Gauteng. The diagram below shows part of a process that occurs during protein synthesis. So already that process cannot be protein synthesis because they're telling you protein synthesis here. But it's another process within protein synthesis, and you should know by now that there are two parts to protein synthesis for us. Okay? Study the diagram and answer the questions. Good. There's a diagram there. Something is happening here with molecule A, and one of these strands is a coding strand. There comes molecule B. Take careful note at the nitrogenous bases that are found in molecule B, and look at the ones in molecule, if I would call this A, strand A, of molecule A. Good. So they say name the specific process. Now guys, what are the clues given in the diagram? The clue, the biggest clue is this guy here, this shape. Do you remember?
Yes.
What's the shape?
That's helix.
Hey! Wow! Sia is not even studying life science anymore. She did life science when? 3 years ago?
Yes.
3 years ago. Wow. So she knows that's helix, but because there are two, we say double helix. Huh? We say double helix. So double helix is only associated with one molecule, and that is DNA. DNA in protein synthesis is only involved in one of the two processes, and that is transcription. So therefore, this process is transcription. Wow, we got it right. Where in the cell does this process occur? If you know that it's transcription, you should know that transcription takes place in the nucleus. Molecule A and B are both nucleic acids. Tabulate two structural differences between these molecules. So let's go back. We've already identified molecule A as DNA. Obviously, the other one, without even looking at the nitrogenous base, we should know if it's the other nucleic acid. There's only two that you studied, DNA and RNA. So this one is RNA, and there's something in this diagram that already tells you that's RNA. But let's look at our table. Tabulate. So first of all, you had to draw a table. To get 1 mark, you need to draw the table. So that's the 1 mark there. And then you compare. As you compare, you notice you're getting 1 mark on either side. On the table, your comparisons must be next to each other. You can't say here a double-stranded molecule and here it has uracil. Right? I don't want to go to the boy and girl thing now. We will leave that for later, yeah? When you get used to me.
Yes.
Okay. We don't want to scare you away so quickly.
I want to come back.
You will come back, eh? So double-stranded molecule, single-stranded. We can see that there already in the diagram. Secondly, the sugar, but they didn't say visible difference. If they said visible, you cannot use the second one. The sugar is deoxyribose in DNA, and in RNA, it's a ribose. Base pairing occurs. No base pairing occurs. Obviously, if it's single-strand, it can't be base pairing. Generally, these two will go together, but we're using them there. Thymine is complementary to adenine. Here, uracil is complementary to adenine. Those are the 3 important differences. We asked for only 2. So, we've given 4 there, sorry. And they asked for 2 structural differences. Reading the question is so important. Not just any. So somebody could have said that one is found only in the nucleus and the other one is found in the nucleus and what have you. But they ask for structural differences, so you stick to that. Describe the role of molecule B in protein synthesis. Molecule B would be the RNA, and that particular one is messenger RNA. What does it do? First of all, messenger RNA copies the code from DNA. transcription, to transcribe, to copy. So messenger RNA, you're naming it, you're getting a mark for that. It forms a copy of the DNA molecule, makes a copy. It copies the code from DNA molecule. And then it leaves the nucleus. It doesn't stop there. They ask you the role of messenger RNA. The messenger RNA doesn't stop in the nucleus. After it makes the code, I'll relax here. No. It moves out of the nucleus and attaches itself to the ribosome, where it provides the code to the transfer RNA molecule. How? It determines the sequence of the transfer. Actually, I would say determines the sequence of the amino acids eventually. How does it do that? By coding for the right transfer RNA molecule. Remember, the messenger RNA is the codon. In other words, if the messenger RNA is UAC, then the transfer RNA this is providing the code now. So we call it the codon. The transfer RNA that comes to attach to this one must be AUG. And this transfer RNA will have amino acid 1 with it. Another one will have an amino acid 2, and so on and so on. So it determines the sequence of amino acids eventually. The transfer RNAs, really speaking, do not determine the sequence. They simply get the sequence from the messenger RNA. We're still with the same question. Use the table. This is a question that gives lots of you problems of protein synthesis. Use the table below to give the sequence of the 3 amino acids that would be coded for by the section of DNA shown in the diagram above. Now we need to go back to the diagram. So if this is the DNA, this is the messenger RNA. So the original code came from the DNA. It formed a messenger RNA. Now what about transfer RNA? Remember this thing that I've taught you when we did DNA, that the whole of protein synthesis. The DNA and the transfer RNA are the same. The DNA and the transfer RNA are the same. Now what do they have here in this here? Can you see what they have here? Anticodon. Anticodon comes from the transfer RNA. So although they're talking about the DNA, you can't take the code as it is there. You've got to convert that code to transfer RNA code, then read this and get it there. So let's go back. So if that is DNA, this is messenger RNA in the middle of the sandwich. On the other side will be the transfer RNA. In other words, as I told you before, you simply rewrite the DNA first. C, A, you will write the T, G, G, C, T, A. But we know that in transfer RNA, there is no T. So we have to take out the T there and replace it by U. Take that T out, U. So the first one is CAA. Go down to the table. CAA is this one here. So the first one is glutamine. The second one is UGG. UGG, yes, UGG. UGG, there you go, is tryptophan. And the last one there is CUA. CUA, CUA, CUA is leucine. And let's see, there you go, glutamine, tryptophan, and leucine. Give the name of the molecule that carries the amino acids in the cytosol. That's easy, the transfer RNA. The bond that forms between 2 amino acid molecules, that is a peptide bond. Notice the ones that are easy, we're not wasting time with. Is the molecule produced by these 3 amino acids considered to be a protein? Explain. We can answer this one too. No, because a protein must have at least 50 or more amino acids. Right now, this molecule has 3, so it's called a tripeptide. Sometimes we may even call it a polypeptide. And we're not going to waste too much of time with the second question. Similar concepts being tested. I went through that one a little slower so that you can see how we get to the final answers. This one, we're going to go a bit slower. The diagram below represents a part of a protein synthesis. This was from KZN, this paper here, this question from KZN. The diagram represents a part of protein synthesis. They want to know, identify the molecules labeled Y and Z. I'm glad that we got these questions because it helped us to look at both both sides of the coin of protein synthesis. But anyway, let's go, and then you'll understand what I'm talking about. So we've got this molecule here. Where is the Y? This one here. Y. And they want Z is this long one here. Okay. So there's one long one there, and there's many of those Y, and this Y is attached to something. Now we should know by now that that has to be transfer RNA, because it's collecting and bringing amino acids and going away and coming back with other amino acids. Other transfer RNAs are coming, and this one that's here is probably on the ribosome, and it has to be messenger RNA. So this is our labels for those two. One would be transfer RNA Y, and Z will be messenger RNA. Name the phase of protein synthesis represented in this diagram. Phase, we'd rather say stage, but phase is fine. Okay. The one we did in the nucleus just now was transcription. So this has to be the other one, and therefore this would be translation. Transcription is a process where messenger RNA makes a copy of the DNA. Copying. Where's the code? I copy the code, I bring it here for you. And translation, we take that code and we decipher that code to something else. We change the code to something we can understand. What can we understand? We can understand what a protein is. So this was in another language. We can't understand encrypted stuff, detective stuff. We are doing some forensics. We come there, we decipher the code. We break down this code. We change this code to something more meaningful to the cell. What is this something meaningful? A protein. Why is it meaningful? Because the cell needs proteins. Okay? So that's what we have there. This is translation. Give the name of the group of 3 bases that are indicated by number 4 on the diagram. In other words, this area. What do we call this here? I told you earlier in the last question, so we're not going to waste too much time there. That would be a codon, because it's on the messenger RNA. It's providing the code. The transfer RNA is breaking the code, so that's the anticodon. Huh? Good? Write down the base codes from left to right that would be found at point 3 on the diagram. Point 3 on the diagram is here. And it's matching with this guy. If that is C, that has to be G. Cute girl. Remember? Cute girl. And with the U, it's A. Normally, it should have been at, but there's no T here. So U is A and U is A. So it's GAA. Guanine, adenine, adenine. And again, our friend, the table comes in. Remember, mindsetters, you do not have to memorize anything on this table. If this question is asked, the table is always provided. You need to be able to read. Here's the amino acid here, the name of the amino acids, and here is the base. Listen this one now. This one, they gave you the base triplet from the DNA template. The last question spoke about the anticodon. Anticodon, you must associate to transfer RNA. So here they're telling you, this is what, if a DNA gives you these things here, these are the amino acids. They want to know, write down the names of the amino acids represented by number 1 and number 5. So we've got to go back to the diagram. Number 1 is this one here, and number 5 is this one here. Right, so if that is GUG, that is the transfer RNA. I told you, if the transfer RNA DNA is generally the same, GUG. But if this is DNA, then just like we took out the T in transfer RNA earlier, we have to take out the U here, and we have to replace it with the T. We don't put A, we replace it with the T, because T and U are the interchangeable ones, not the ones that combine with each other. That's what you must understand. So GTG is the one, that is number 5, and what's the 1? This is the one here. So this is UGG. Now they gave you the messenger RNA. So the DNA would be the opposite of this, because messenger RNA stole the code from DNA. So where the U comes in, now here you'll put A, because this is what it would have combined with. Okay, and that's what would have been there as well. So can you see it's the same still? And if that's G, that would have been C, this is also C. If that's G, that's C, this is also C. So your messenger RNA is the opposite of both DNA and transfer RNA. The difference between the DNA and the transfer RNA is the letter U and the letter T. That's what you need to keep in your mind. So we are looking for ACC and GTG. ACC and GTG. ACC, there we go. ACC, that's one there. Threonine, and GTG, valine. There we go. There we go. Okay? We have answered that one successfully. Good. So that's how protein synthesis taken care of. DNA, if you need to look at DNA again, remember what can they ask you in DNA? The structure of DNA. They can give you one DNA, you must complete it. They can ask you about the labels of the different things on DNA, the functions of DNA, and DNA replication. And remember, whether they ask you here or they ask you later, you can talk about DNA fingerprinting. They can ask you, it falls under in our syllabus now with genetics, but remember, they're both in the same paper, so it doesn't really matter. So DNA fingerprinting is also important, so you need to know that.
I think it's time for a break now, so don't go away. We'll be right back. Welcome back from the break. Let's not waste any more time and get straight back to today's revision lesson.
We're now moving on to questions on meiosis. We can try and do both the questions of meiosis here. The following diagrams show chromosomes as they might appear during various stages of meiosis 1 and 2, starting with a cell pair of homologous chromosomes. So they're giving you these diagrams, A, B, C, D, E, F. Give the letters of the drawings that represent meiosis 1. Before we do that, in the correct order in which the phases occur. There's your diagrams. First of all, we need to determine which are meiosis 1 and which are meiosis 2. This one here is definitely meiosis 1. Why? Because we can see crossing over taking place there. This one above, look at the chromosomes. The chromosomes have two strands. They have chromatids, both of them together intact still. They haven't separated. So this will also be 1. In this one, look clearly. Single strands, chromatids have separated, so this can't be 1, so we leave that one out. This one also, chromatids have separated and moving apart, so it can't be that one either. Here, it's one chromosome on the equatorial plane on its own, so that also can't be. This one, the chromosomes are lying next to each other, homologous chromosomes, that's also 1. Now, the question is, what is the order? And the order would be C. First, the chromosomes are lying next to each other. Then crossing over D is taking place. And this would be anaphase 1. Anaphase 1, why? Because the chromosomes have separated and they're moving to opposite poles of the cell. So CDA. So two skills here. The first skill you had to know, or to put into practice, was to identify which is meiosis 1 and which is meiosis 2. The second skill was to put those ones that you've identified as meiosis 1 in the correct order. So you have to know your marbles. You can't take chances here. You can't guess here. State one visible difference between the events in meiosis represented in diagrams A and E. That's A, what's happening there is anaphase 1, and this is anaphase 2. By now, without looking at the diagrams, you could have said so. But they ask you specifically only the differences between these two phases as you can see them there. And they are, the whole chromosomes are separating in A, and in E, chromatids are separating. You can't see the centromere splitting. You can't see that there. You cannot even see invagination taking place or a cell plate forming. You can't see that. You can only see what they have there. Good? Name the process in meiosis represented in the phase shown in diagram D. What is the process? Notice they're asking you, not the phase. They want to know the process shown in this diagram here, D. And we spoke about it earlier, so we're not going to waste your time, and that would be crossing over. Notice the difference between the process and the phase. If they ask for the phase, that would be prophase 1. But they did not ask you for that. They asked you for the process that's happening in that phase, and that is crossing over. Understand the difference. Explain why the process named in question 3.3 is biologically significant. Why is crossing over? No matter how they ask this question, what is the significance of crossing over? What is the significance of what you can see in a diagram? The answer is always going to be the same. What does crossing over do? It allows for exchange of genetic material, which brings about variety in organisms. So it introduced, now this one, the answer is put the other way. It's giving you the end first. It introduces genetic variation. Why? Okay, and it's giving you other stories which ensure that the offspring are different to the parents. That's already in genetic variation. I would still give a mark for because it allows for exchange of genetic material. You must add that in your answer, that it allows for exchange of genetic material, which brings about genetic variation in a 3-mark question, and you can add this last part. This can, it may, it's not 100%, but it may increase the chances of surviving because there's a variety available. Good? An organism has 20 chromosomes in its body cells. After meiosis, the gametes produced contain 11 or 9 chromosomes. It had 20. Getting 11 or 9. Explain the events in meiosis 1 that resulted in the gametes having different numbers of chromosomes. That's easy. It's non-disjunction. It means some of the chromosomes did not separate, and therefore one had 11 and the other one had 9. When? During anaphase 1. Or you can say anaphase, where one pair of the chromosome failed to separate, resulting in 11 in the one and 9 in the other. So therefore the one cell has one more and the one cell has one less. Remember, you haven't learned this. You've learned trisomy 21, Down syndrome. But it's non-disjunction. So this is an application question of the same topic. You've learned it, this non-disjunction. So you must be able to apply it to a different situation without giving what is this disease. You don't know what this disease is or whatever. You don't know what organism is involved. But you know that it can only happen because of non-disjunction during anaphase of meiosis. And therefore one has one more and one will have one less. Good? Move to the other question now from Gauteng. The following diagram shows a cell undergoing meiosis. Study the diagram and answer the questions that follow. Here's a beautifully drawn diagram there. Some shading and what have you. And there's only 2 simple labels, A and B. And this A is the one that you normally get wrong. Because the minute you see A, you start labeling that as nuclear membrane. We'll talk about it just now. Okay, we'll have to talk about it now anyway. Are you with us, Tilsia?
I'm still with you.
You sure?
Yes.
I'm not boring you, am I?
No.
Are you sure?
I'm just getting mad at myself when I'm not getting the answers because I'm like, I know the stuff.
You know the stuff because now I'm going to test you just now.
Oh, God.
You better start Googling there quickly. Provide labels for A and B. So A, I said, can't be the nuclear membrane. So you think you can try that one? A. If it's not the nuclear membrane, what membrane is it?
I'm sorry.
You can't, eh? It's too far back.
No, it's too
Well,
far
if it's not the nuclear membrane, it's the cell membrane.
Oh,
Remember,
yeah.
guys, remember, Sia, remember, people. When the cell starts dividing, the nuclear membrane disintegrates. In some diagrams, you may see something like this in the early stages where you see the chromatin network visible and so on, and you see this dotted line. That would be the nuclear membrane. But remember, after a while, that's going to disintegrate. It's not going to be there anymore because there's no place for all these other things when the cell is dividing. So you're only seeing nuclear material. All the other organelles have disintegrated and so on. So that has to be the cell membrane. And B is easy. It's the spindle fiber. Does the cell shown above represent a plant cell or an animal cell? First, you have to look at it. Is the plant cell or animal cell? You would say you should have said it's an animal cell. And give one visible reason for your answer. If you look, especially in this area here, you will see that the centrioles have formed the spindle fiber. In a plant cell, the cell wall would have formed the spindle fiber. So that's where our answer comes from, originates. Now, the one is that there are centrioles present and only animal cells have centrioles, or there is no cell wall visible and animal cells do not have cell walls, plant cells have cell walls. Remember when you're giving differences also, try to say, instead of saying it does not have a cell wall, say that this cell has as an outer covering a cell membrane, a positive reason. Whereas plant cells have as their outer covering a cell wall. Try to keep your answers positive rather than negative, unless there's no other way of making it positive. Okay. What phase of meiosis is represented in a diagram? Sia, you think you can manage?
I forgot the phases. It's ana something,
Okay,
is it not?
interphase, prophase, metaphase, anaphase, and telophase. Now
I'd
think
say
of where
anaphase.
these chromosomes are situated. Where are they in the cell?
In the center.
They're in the center or the middle or the equator, right?
Metaphase.
Ah, there you go. You see? A little bit of jogging the memory and it will come back to you. All back. Why? Because you probably understood the work. You did not swat it when you were in school. I'm so happy.
didn't
I found
fail.
somebody that understands rather than swats. I've been singing that song all day today. Metaphase 2. But why metaphase 2? Because they are in the middle, that's metaphase. But why metaphase 2 and not 1? Because they're lying singly. Don't think that you're seeing two chromosomes, so they're lying together. They're not on the same spindle fiber. They are independently on different spindle fibers. So that means these two are singly there. Their partners would have been next to them, besides them. When they're together, it would be metaphase 1. Now it's metaphase 2. Explain your answer. Well, Kano, you gave them the answer already. Because single or unpaired chromosomes are aligned on the equator. Wow, we've done that one too. We're still busy with that one. Give the diploid number of chromosomes for this cell. Now, how many are here? This is metaphase 2. So there were 2. That means the chromosomes have already separated. So originally, what do we have here? We would have had 4, diploid. Draw the cell in the next phase of meiosis using shading to show genetic variety. So you're showing the next phase, anaphase 2. Why did they say use shading? You need to show these differences there. So they gave you marks for all of these things, for drawing that's the right phase, and for the shading that's done correctly as well. So you know what to do there. Abnormal meiosis can produce abnormal number of chromosomes. For example, Down syndrome, where gamete with an extra chromosome fertilized by normal, and it will have 47 instead of 46. On which chromosome number will there be an extra? We spoke about it just now, chromosome number 21. Explain how this normal was formed in the gamete, similar to what we explained earlier. During oogenesis or spermatogenesis, the chromosomes fail to separate because they asked you here in the gamete. So you have to talk about that. But remember, meiosis only takes place for gametes as well. State Mendel's principle of segregation, and Mendel's law of segregation states that for each characteristic, there are 2 factors. And these 2 factors separate during gamete formation, enabling a gamete to contain only 1. So sperm will only have 1 of the factor. The egg will have the other one. When they come back, the individual will have 2. Phylogenetic tree below shows one interpretation of origin of humans. The dotted lines indicate the possible evolutionary relationships, and the vertical bars show the period during which the organisms are believed to have existed on Earth. All right? So they're telling you here, we're going up. It's telling you when they lived. 1 million years ago, 2 million years, 3 million years, 4 million years ago. All right? And they're showing you possible relationships as well. So obviously, homework, guys, you need to look at this diagram carefully, obviously. You need to see that. Phylogenetic tree, another name for it, by the way, just as a matter of interest, anybody? Another name for a phylogenetic tree? We call it some diagram with a C. We call it a? Hey, Looney, we are in trouble today.
We gon' win.
Nobody go win yet. I think we go win.
We gon' win.
We go win, yeah. A cladogram or a clad diagram is another name for phylogenetic tree. Do not use the word family tree. You will not get a mark for that if they ask you in a one-word answers. It's either phylogenetic tree or a clade diagram. Okay, so that's the story there. They want to know, use a diagram to identify one organism that may have competed with Homo heidelbergensis for resources. All right? So we need to look at the diagram, and we need to look at when will an organism compete with another organism? They live in the same period of time. And what would you think in this particular case? Which one could have competed with them?
That would be Homo erectus.
Sorry? Homo? Neanderthalensis. Neanderthalensis. Okay? I'm hoping Hamida is watching today because she made a meal of the other time I was talking about Neanderthalensis. So I practiced the whole week about Neanderthalensis. Huh? I'm getting it right now, I think. Hamida and her daughter had a good laugh at my expense. So we are saying that is that, but you are both wrong, so you get no points. It was Homo erectus. Okay? Why? Let's go back to this thing here. All right? There, look at that period, and look at where they are. Homo erectus is the one that's there, and if you can look further, you'll see the same things further as well. Okay? Identify the common ancestor that gave rise to both Paranthropus and Homo. So Paranthropus and Homo, we must look for them on the stuff here. Where's Paranthropus? There we go there, right? Paranthropus is around there. Where did this Paranthropus come from?
It came from there.
Where is the, and what did you say? And Homo, isn't it?
Yes.
All right. So which one is a common ancestor for that one and
It's
the Homo?
Australopithecus.
Is it this one here?
No.
Can't be that one. Why not
Below
this
that one.
Why is it not this one? Remember, this one I'm giving guys practice now, right?
Yes.
Later we're going to ask you questions like one at a time, so you're going to get points. Now we're just having practice so you can start talking. Why is it not this one?
That one gave rise to
Today.
both para.
The Paranthropus.
Yes.
Did it give rise to Homo?
No.
No, so therefore it can't be a common. Common means it would have been linked to both of them.
Yes.
Okay? So where do we go to them?
Down.
There,
Down.
isn't it?
Yes.
Can you see here? The branch is going that way, and eventually to the Homo species there, and from there to the Paranthropus there. Good? So Australopithecus afarensis. And I told you afar is a place. It not mean far away, no? And there we got that one there. Good, so we got that one right. For what period of time did Australopithecus africanus exist on Earth? Show all working. I'm going to go straight to the answer here. We don't want to waste time for that. So we look at the time from 3 million years minus 2.4 million, and the answer would be 0.6 million or around there, depending what you read there. Remember, they've given you a 1 point difference there.
Yes.
Okay, so if you work it out with 2.4. Now, if we mark in an exam, you can't say 3 minus 2.4 and get 2 and then say, but that was also in the answer. you can't get that, right? Okay? That doesn't work. If you have that, you must have the whole thing there, or can see where the O is.
Yes.
Either or. Okay? Name one piece of evidence that could be used to prove that Afrikaners existed during the time period calculated in 1.3. One piece of evidence that could be used to give that there. Oh yeah, they want. You don't need the graph there. What? How would we know that it was living in that time?
We can use relative dating or
Yeah. So in other words, what we're looking at
the paleontology.
what we're looking at the paleontology. What do they look at?
They look at the
Fossils. Yeah?
fossils.
So we look at the fossils and we date the fossils.
Fossils.
We can either use a relative, and the other one is a radio, carbon, or active dating. Good. We're getting there, guys. So, but the main thing is we are looking at the fossils. The evidence is the fossil, but we date it. Which organism, Homo ergaster or Homo neanderthalensis, is more closely related to modern day humans? Now let's go back there. So we look at ergaster and Neanderthal.
It will be
All right. Modern day humans is
Homo sapiens.
sapiens. Let's do that. That's modern day. So which one? They ask for ergaster, this guy here, or Neanderthalensis. Which one is closer?
Neanderthal.
Obvious, because it is a branch. They have one common there, the Heidelberg genus, that is the common ancestor, whereas Homo ergaster comes from another tree.
Yes.
In other words, it's like taking your family and asking you the question, mindsetters, if you have two brothers from one father, and then the father's brother's son, who is closer, the two brothers from the one father, or are they closer to their cousin? Obviously, the two that came from the one father there, they are closer than the one that came from the two brothers on the top, because they came from the same branch. This is what we're saying. So it's similar to a family tree in real terms, that you need to read it. So you have to follow the dotted lines in this particular case. All right. Right, there we go. Explain your answer to question 1 using information in a diagram. And there's the answer there. Homo sapiens and Neanderthalensis share a common ancestor. I would like to see what they put there. Or we can say both of them evolved from Heidelbergensis there, in that case there.
Okay, guys, I hope you are enjoying this revision session. If you are struggling and need help, please post on the page or send your questions to the Learn Extra Help Desk. The links are on our Facebook page. But now it's time to take a very quick break. Don't go away. We'll be right back. Welcome back from the break, guys. Doing revision is really important, so I hope you are enjoying this lesson. I'd love to chat to you on our Facebook page or on Twitter, so please let us know how you are doing. Enough of the chat, now it's time to get back to some more revision.
Study diagram below showing structures of different organisms. So they're giving you a butterfly wing, human limb, whale limb, and a bat wing. Which diagram represents, now we're starting with the teams. Guys, our mindset is just so that you know what we're doing. We're passing a question to the blue team. Blue, no? And red. We give the blue team first the question, and then the next question goes to the red team, and so on. And we'll be scoring them as we go along. At the end, we'll see who has won, who is the weakest link. Right. Okay, so that's how we're going to do this. If the blue team doesn't answer, whichever team, they don't answer their question, we pass it on to the other team as well. So we have that as well in our stuff. Remember, guys, you've got to pick up the mic while you're talking, right? Which diagram represents a terrestrial animal? You've got to listen carefully to this question. I put that word there. It was not in the question. I put it in deliberately. That's a terrestrial animal with a structure that is homologous to the structure in diagram 4. What is diagram 4? Diagram 4 is a bat wing. Now, so it can't be 4 because we're talking about 4. We need from 1, 2, and 3 a terrestrial organism that is homologous to diagram 4. Quickly.
It's diagram 2, the human limb.
Diagram 2, the human limb, and that is correct, so you get 1 point. We can also say 3 in this case here, but why can't we say 3 in this particular case?
It's because terrestrial animals are animals that live on land.
There you go. Hey, I should give you a bonus point for that one there. Somewhere along the line, we're going to skip your points. Okay, so you got 1 point. You better keep it somewhere. All right, so it can't be 3 because we asked for a terrestrial animal and not any one of them. So you be careful there. The whale is not a terrestrial animal. What information, Team Red, we're going to win. What information do homologous structures provide about evolution? What does homologous structures tell us about evolution?
They provide information that species with similar structures, they evolve from common ancestors.
Excellent. They're talking about common ancestry. Hey,
No.
Vugile.
They were just acting.
That's
the real
They were
deal.
acting. Now the real thing has come out. Okay. So scientists are suggesting that homologous structures show that because they're similar structures, they show that two organisms have developed from a common ancestor. Now, I need to clear up something that we spoke about in a previous show when I was doing evolution. I discussed homologous structures, I discussed analogous structures, and I discussed vestigial structures. Homologous, CAPS only prescribes homologous structures, not analogous, not vestigial. I added analogous and vestigial to provide a comparison so that you can understand the difference. Analogous structures do not support evolution because there are different structures. For example, in this particular case, that would be the bat wing and the butterfly wing. They're both wings. They both serve the same function. But if you look at the structure, they are different. This one has bones, this one has membranes. So this suggests that they do not have a common ancestor. But you do not have to know analogous structures, neither do you have to know vestigial structures. So remember that you only worry about homologous structures. And while you're worried about that, don't confuse homologous structures with homologous chromosomes in meiosis. Good, so far we are even, right? So it's easy to keep score. All right, so that was done there. By the way, that section dealt with evidence for evolution. That is what evolutionists use to say that evolution has taken place. They proved that by showing homologous structures. There are other evidences that you must study. I'm not going to go into all of that now. We took a sample of a question. The graph below shows the results of an experiment done to investigate the average time that fruit flies can survive without food. It's called starvation resistance. The average starvation resistance time is when 80% of the population has died out. The researcher placed 5,000 fruit fly offspring from the same generation in a large container without food, and they recorded it. The eggs, blah, 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 the average starvation resistance was recorded and repeated until the 8th generation. Identify the dependent variable in this investigation. Blues. Aye?
The fruit fly.
Fruit fly. No.
Starvation of the fruit, the starvation.
The starvation resistance is correct. So our red team is one up now because the fruit flies are the independent variable. You changing them. In fact, they're not even an independent variable. It's the amount of food would be the independent variable. Remember, the fruit fly is what we're investigating. But what are we investigating? About the food, isn't it? We're changing. What are we changing? We're changing the food. And if we're changing the food, that means that's the independent variable. And when we change the food, some will survive and some won't survive. And that tells us about their resistance. That we can't control. So that is the dependent variable. And guys, remember, although your answer is incorrect, you're not going to die because of that. The most important thing about that is everybody out there is going to learn from your mistake. So although you're not getting a point, you're getting your point across. All right. Okay, state two factors that should have been kept constant during this investigation, because the question was asked first to you and it was your question. Now it's their turn. Red team.
The factors that should be kept constant is the environment
The environment must be kept constant.
and
Good.
the type of species used.
The
the
type
fruit
of
flies.
fruit flies used, you gave us two, and there's obviously more than that. We can talk about the temperature, the flies that must be the same generation or the age, the flies of the same species, there's your one, same amount of food given, number of eggs used, and the environment there would be that story that you're giving. The environment would be covered in temperature. But remember, you must be more specific. But you get the 1 out of 2 there. Good. We move to another question now, or the same question, sorry. Now they're giving you the results. All right? In the first 8 generation there. So look at what has happened. Over generations, something has happened, and you can see it clearly. Da 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 da. Okay, so if we drew a line graph, it would look something like that. Okay? Are we happy with it? Good? Long question. In terms of natural selection, explain why the average starvation resistance time of the fruit flies is different in each generation. Less, more, most. Why? In terms of? Anybody?
Natural selection is about adapting to your environment.
I will
in
kick
which
you hard. Don't use that word.
Okay.
Adapting.
Yeah.
Right.
So the first lot of flies went. What's this?
First, what must you say in natural selection? First thing, there is
Large variety.
Yeah.
variety. Okay, so in this particular cycle, there were some that had higher resistance and some that had lower resistance. I'll carry on now.
Yeah. And so as they died out, or some
Which ones will die out?
the ones that are less
Less resistant.
yes, sir.
And
then the ones that are more will survive.
And what
in
will they
which,
do?
In which they will like not adapt.
no, they can't adapt. They
Yeah.
either are or not, right?
Yes.
But what can they do if they survive?
They will-
Come, you like that one?
They will utilize the one that
No, no, no.
They will put-
Help
They
them.
will reproduce.
That's, you see, I know you only think about reproduction. I catch you. I know how to catch you guys. So they will reproduce,
is
right? And therefore, over time, there will be more that are resistant and less that are not resistant. Now, before we answer this question, mindsetters, just to remind you, we had some words that we needed to remember. We must start with variety or variation. We must go towards competition and not adaptation but suitability. Remember, when we talk about suitability, it means that some have it and some don't have it. They don't adapt. Adapt means they became like that. And that is Lamarck. That's what Lamarck said. Okay? Suitability and then survival. And with survival goes reproduction and what else? And this is known as natural selection. And this over several generations. This is what we need to include in the answer. But we can't write those words and expect all the marks. It's not going to happen. You need to contextualize it. So first of all, we said, and here's where your answer is coming, there's a genetic variation in a fruit fly population. Some had longer starvation. You must specify. When you're saying variation, you must specify what is that variation. Some can survive longer and others not. So you dealt with first bullet. When there was no food, there was competition for that. So the fruit flies with shorter starvation resistance did not survive. So they were not
Suitability.
suitable.
Yes.
While the ones with longer starvation resistance with time survived. Why? Because they were more suitable and they survived and therefore they could reproduce with longer resistance time. Genes for the favorable trait were passed on to subsequent generations. We must also talk about that. Genes are passed on, not characteristic. The gene for the characteristic is passed on. Are we okay with it, guys?
Yes.
So can you see that with any natural selection question, if you take this and adapt it to the situation, you'll be fine. Are we happy, guys?
Yes.
Anybody has a problem? I love this. I love this. I love this. This is getting right. So that's. Remember, there's another section in the first part of evolution, and that is speciation. We haven't discussed it. We haven't put a question there. But speciation, and the only one you must know is speciation by geographical isolation, or books may also, older books will call it allopatric speciation. Sympatric speciation is not prescribed for the syllabus. You must only concentrate on allopathic, where there's a barrier. All right? Okay. Now, we're going to genetics and we're going for the kill first. We're not going to go to the simple ones. We're going straight to the heavy ones. Right? They're telling you about blood groups. I'm not going to read the whole stories there. The blood groups of individuals, WX, are not indicated there, right? Okay? They're telling you that this is blood group O, B, A, O. This guy, they don't know. That guy, they don't know. Which one from here, group red, nah? Group blue, sorry. Which one can we definitely say what the genotype is? Is it O, is it A, B, or O? Which one can we say definitely this is the genotype without hesitation? And what is the genotype?
I think it will be blood group O. It will be blood group O.
It will be blood group O. And what is the genotype for blood group O?
It's
How do we write the genotype? If we're working out the problem.
It's recessive.
So therefore it will be two
I.
small
eyes.
eyes. Because it's recessive, good word you use, because it's recessive, it can only exist in the homozygous condition. So that's guaranteed. And once we got that, we will know that this individual must have one small eye, isn't it?
Heterozygous.
Because it's getting it from the mother. And this one too must also get one small I. Are we all clear with that one?
Yes.
Are we answering the question yet? No.
No.
We're doing homework.
Homework.
Whenever you do genetics, you do homework first, because that's going to help you. We still don't know what is W, and we don't know what is X. But we know that that is two small I. So what does that tell you about X? It has to have
I.
one small I. All right? Are we all clear?
Yes.
And what can we say that this one had to have one small I?
No.
No, we can't say that. What can we say that it had to have?
AB.
It is
It had to
heterozygous
have-
A and B.
B
And
and A.
A.
So already gave you the answer.
Yes.
Yes.
Because why A there and B there? All right? Are we happy, guys?
Yes.
Can you see how you can do the homework?
Yes.
And this is how you're going to solve pedigree problems. Write down all the possible genotypes for the individuals W and X. I can't ask you because we've done it already.
Yes.
Okay? Let's just open it up for the mindsetters. Now remember, you don't write A, B like that. You have to write it like this. W is IA, IA, IB. That's how you write for AB. And for A, it's either IA, small I, or it can be IA, IA. But in this case, it had to be that one there. Okay? Why we got so many by L or by X? All right, we come to X's now. Okay, let's go to X again now. X, the child is O. So X could have been O also, isn't it? Because that and that will give you this. X could also be A, but it had to be heterozygous A to give that one small I. Or it could have also been B, but heterozygous to give that one I. Are you catching that?
Yes.
All right, so that covers us on the blood group. I'm not going to waste time with the rest. What is the probability or chance that they will have a child who, oh, sorry, where the hell am I? All right. A rare form of rickets in humans is caused by a sex-linked dominant allele. Listen very carefully. Number one, clue one, sex-linked. Clue two, dominant allele, R, which is carried on the X chromosome. An unaffected female married an affected male. Okay? So unaffected, in this case, team red, no? What will be the genotype of this unaffected female? Remember, you got to use either capital R or small r. Two small r's, isn't it?
Yes.
Because this is dominant.
Dominant.
But we can't just write it like that. Because she's a female, we have to write it X small r, X small r. Are we clear with it, guys?
Yes.
Both of you guys. Right. And the male is affected. So he is X, Y. It's easy. What must we put by the X?
Capital letter R.
Capital R. Do we put anything on the Y? No. No, because no gene is carried on the Y. Y is too small to carry any genes. Are we clear with that one?
Yes.
So this is what we do only when we're dealing with sex-linked genes. We use X and Y with superscript for the letters. What is the probability that these two now will have a child who will be an affected female? 50%? Remember, she's getting this R from the father. And remember that this is dominant. What do you say? 50 also?
25.
25. You're both wrong, right? Okay? You're right,
Yeah,
but in a
right.
way, right? 50%, okay? X are there. In fact, I'm thinking this is wrong.
It's right.
I just want to work something out quickly in my head. X R. See, the father can only give capital R.
It is.
Mm.
And it's dominant. So then, the daughters, where do they get the X from, the daughters? One from the father and one from the mother. Right. So from the mother, they're going to get the small R. Hey, don't move around. They're going to get a small R, isn't it?
Yes, 50.
But from the father, they can only get,
Capital R.
so that means,
5% chance now.
because it's dominant. Look at their children. Their children will all be XR,
Yes.
XR.
Yes.
So they're all going to be that way. He doesn't have a small r. So he's only giving the capital R. Are you seeing that?
Yes.
Right, so that's wrong. So it's 100%. What is the probability chance that they will have a child who will be unaffected male. Now,
Zero.
male is slightly different, right? Because
It's
the
100
male is getting the X from who?
from the mother.
From the mother. Because if the male gets the X from the father, then he's not a male, isn't he? Right? So it's a female. So XR is getting from the mother, and he gets the Y. Right?
Yes,
So
it's 0.
what did they ask for? Unaffected. And the answer is
100%.
100%.
100% as well. Here it's saying 0, they're talking crap. It's 100%, right? Should be, I missed out the thing there, 100%. Are we happy there?
Yes.
Explain why this disorder, although it is sex-linked, does not affect males only.
Because it is found on, it is a dominant. found on-
Dominant, that's all, stop there. Because it's dominant.
Dominant
If
and
it was
found on the X
recessive,
chromosome.
then it affects males quicker. Okay?
Yes.
Good? Dominant can affect both there. Good. Excellent.
We've come to the end of today's show. Thanks for joining me and for chatting to me during the show. Please visit our website learn.mindset.co.za to get the notes and to watch the videos from earlier in the year. And remember, we are here to get you through the year, so stay connected. Goodbye.